Now we're going to talk about individuals who are resistant to infection or who can regulate infection such that they can live with the virus quite easily. So let's talk about these two genetic reasons why some individuals can tolerate HIV exposure and infection. So the first set of individuals are resistant to infection, specifically the macrophage tropic virus. So we learned in a previous video that initial infection by HIV, which is a sexually transmitted disease, usually occurs via the macrophage tropic version of the virus, attaching to CD4 and CCR5 proteins on the surface of macrophages and dendritic cells, usually within the reproductive system. So there are individuals who inherit a certain version or allele of CCR5, which allows them to be resistant to infection. So the CCR5 gene makes a protein that's found on the surface of a number of immune cells, including macrophages and dendritic cells. It doesn't play a huge role in the immune system, and we're actually not going to talk about its function within the immune system. What we are going to talk about is the fact that there's a version or an allele of CCR5 in the human population called delta 32. It has a deletion in the gene that ends up producing a non-functional protein. So some individuals who inherit this mutant allele will make defective protein. It won't go to the cell surface. So you could be heterozygous for this allele, or you could be homozygous for this allele. Now, the individuals who are homozygous for this allele, this delta 32 allele, basically don't make the CCR5 protein. And their immune systems seem to be fairly normal. And what the bonus that they get is that they are resistant to infection by the macrophage tropic virus. So individuals who have this genotype, being homozygous for the CCR5 delta 32 allele, are resistant to infection, specifically by the macrophage tropic virus. Their cells lack CCR5, and so individuals who have this phenotype have been repeatedly exposed to the virus and not infected by the virus. So those individuals are resistant to infection. They could be infected by the lymphocyte tropic virus. So if they had a blood transfusion, which then the lymphocyte tropic virus were in there, then that would attach to CCR, the CD4 protein on the surface of CD4 T cells, as well as that CXCR protein found on the surface of T cells as well. Okay, so those are some individuals who are resistant to infection, and there you go. They wouldn't get the initial infection. There are another population of humans who are called long-term non-progressors or elite controllers. These individuals can be infected by HIV, but their immune system does a really good job of keeping the virus levels so low that they have no immunodeficiency after infection. So they control the infection and they do not progress to AIDS. So how does this occur? Well, let's go back and remember that the immune system has a way to detect virally infected cells and deal with them. And that involves presentation of MHC class 1 molecules. So if a cell is infected with a virus, we know the pathogens, its proteins are degraded by the proteasome, sent into the ER, loaded onto MHC class 1s, and presented to CD8 T cells. And we know that MHC class 1 molecules, the alpha chain of those proteins, are generated from genes such as HLA-A, HLA-B, and HLA-C. And each allele or allotype will present different peptides based on their peptide binding motif. Now, most individuals, most humans, do a really poor job of antigen presentation of HIV peptides. So in the human population, there are not a lot of good alleles that bind HIV peptides and present them to T cells. But there are some good alleles which individuals who have these alleles actually have a much better time presenting peptides and therefore controlling the infection. So two alleles that have been discovered and well characterized are the B57 and the B27 alleles of HLA-B gene. So individuals who inherit those alleles present on peptides on their B27 isoform or B57 isoform. And those isoforms do a really good job of loading and presenting HIV peptides. So if an individual inherits these alleles, one or both of them, then they're presenting peptides to CD8 T cells, and those cells are killing virally infected cells. Do they kill all of them? They do not, but they kill a lot of them, so much so that they can control the infection. So in individuals who inherit these alleles, they have low levels of virus and fairly normal CD4 positive T cell function. So again, these individuals are called long-term non-progressors or leak controllers, and that term refers to the fact that they are chronically infected with the virus, but they do not progress to immunodeficiency or AIDS. They control the infection, and it has been established that this control is due most directly to the inheritance of certain alleles of HLAs, specifically HLA 57 and 27, for example. And that allows for excellent antigen presentation of HIV peptides. So these are two examples of individuals whose genetics might lead them to either resist or control HIV infection.